I have already discussed the structure of bacteriophage in my previous lesson. In this lesson, we are going to study about the life cycle of bacteriophage. Welcome learners, I am your teacher Dr. Anam. Let's talk about the life cycle of bacteriophage. The first step involved in the replication of bacteriophage is the injection of its genetic material into bacterial cell. The steps involved in this process are landing. In the process of landing, the bacteriophage, it lands on the surface of the bacteria. The second step is attachment. Bacteriophage is attaches to the surface of the bacterial cell wall at a specific receptor site by the help of their tail fibers. This process is also called absorption. This attachment is the courtesy of weak chemical bonds which are formed between the receptor sites and the bacteriophage. In the third step, viral genome is injected into the bacterial cell. And how this is happening, that first of all, the bacteriophage, it will release the enzyme lysozyme, which is an antibacterial enzyme, and it digests the small parts of cell wall. After that, a rigid tube is propelled out of the sheet to puncture the hole in the bacterial cell wall. After the hole is produced, the genetic material, it moves from this capsid through this tube and it enters into the bacterial cell. Now all these steps will be the same for all the cycles of a bacteriophage. After the entry of genetic material into the bacterial cells, there are three phases. One is lytic cycle, lysogenic cycle, or induction. Let's study one by one. The lytic cycle. Here the word lytic means breakdown. It means in the lytic cycle, the breakdown of bacterial cells takes place. Here is a diagram of lytic cycle. As I told you that the first three steps are the same for every cycle. Here the bacteriophage, it lands, then it attaches, and then viral genome is penetrated into the bacterial cell. After that, DNA and protein synthesis, it takes place. And how it's going to take place, that it will hijack the biosynthetic machinery of bacterial cell. Viral genome which enters into the bacterial cell will hijack or take over the machinery of bacteria and use that machinery for the synthesis of their own DNA, protein, and tail fibers. The next step is the assembly of phage. The new genetic material which is formed is actually packed into the head, or which is capsid here. What will happen, first of all, the DNA material is packed into this capsid, and after that the tail fibers, they are attached like this. And in this way, the whole bacteriophage is constructed inside the bacterial cell. Usually 25 minutes are required for the synthesis of 200 new bacteriophages. So this small amount of time is required to make so many new bacteriophages. The last step in the lytic cycle is the lysis of bacterial cell. As the number of bacteriophages starts to increase inside the bacterial cell, it pushes pressure on the cell wall or weakens it, and eventually the bacterial cell wall, it will rupture and all the bacteriophages, they will move out of the bacterial cell. At this stage, these phages or these bacteriophages, they are actually called lytic phage. The second life cycle of bacteriophage is lysogenic. Lysogenic word is made up of two words, lyso and genic. Lyso mean to lose, and genic mean to produce or to generate something. In other words, lysogenic cycle involves the production of such bacteriophages that loses their ability to break down the bacterial cells. So in the lysogenic cycle, no bursting of bacterial cells takes place and bacteria keeps on living its life normally. So what happened after landing, attachment, and penetration of viral DNA? What happened after landing, attachment, and penetration of viral genome into the bacterial cells? It incorporates into the genomic material of bacteria. Instead of hijacking the machinery of bacterial cells, it incorporates into the bacterial genome. As it is incorporated into the genome, It will not replicate or synthesize its DNA or protein material. Now, the size of the viral genome is so small that it will not cause any harm to the bacterial cell, and the cells of bacteria, they will keep on living normally. And what will happen, that this viral material, it will be transferred to the daughter cells or new cells of bacteria as the bacterial cell keeps on living. So it will keep on passing the viral genome from the parent cells to the daughter cells. So that was the lysogenic cycle. The third life cycle of bacteriophage is induction. The word induction means action of bringing about or giving rise to something. So in induction life cycle, the lysogenic cycle is actually converted into the lytic cycle. It means that induction give rise to the bacteriophages that can cause lytic cycle or lysis of bacterial cells. Now, what will happen in the induction life cycle, that viral genome which was incorporated into the bacterial cell, it will detach itself from the bacterial genome, and after that it will hijack the bacterial machinery and will start producing its own DNA and protein. After that, what will happen, that assembly of phage will takes place, new cells of bacteriophage will form, and eventually the lysis of bacterial cell will takes place. So that was all about the life cycle of bacteriophage. If you have any question, you can ask me. If you like this video, give it a big thumbs up, subscribe to my channel. I will see you in the next video.